I will explain you the sample techniques or the sampling techniques to analyze and measure biodiversity. There are different sampling techniques. So let's start to analyze with the terrestrial vertebrates. So the first thing that is very important to know is the species target. So if you are studying rare species, then you will follow one kind of technique, and if you are studying any kind of a common species, then you will follow another kind of a technique. The second thing you need to know is the taxonomy and the classification of the species. It means that if you want to study biodiversity, you need to identify species. If you cannot do it directly, you can ask some expert to do it for you. There are different methods for sampling of terrestrial vertebrates. There are direct observation or indirect observation when sampling terrestrial vertebrates. In the direct observation, we use our naked eyes, binoculars, or other equipment. In the indirect observation, you just follow tracks you recognize, footprints, songs, calls, etc. There are standardized sampling methods that can be used for, like trapping, for instance. You don't have to use a killing trap for small mammals. You can use pitfall traps. Now, pitfall traps are traps that are hidden in the ground and animals just fall down. For example, if you want to sample birds and bugs, usually direct observation methods are adopted. Sometimes using net for observation can also be used. So it means that nets are building or some special places that are can be used for random sampling. For instance, you can collect the species inside the nets, and then you identify them and release them back to the normal habitat. There are other methods for sampling of mammals. For rodents, for instance, using adhesive or sampling tubes that just collect the hairs from the body of the animals, and by looking at the hairs, you can recognize the animals. Or for instance, we can use the howling and calls. In this case, we just send a howling of a specific species, such as of the wolf, for instance, via a sound device, and they will recognize their call. For tortoise, we can use traps. For birds of prey, for instance, we can use to recognize the pellets and bones that are inside these pellets. Now, this all are indirect methods that's very useful to identify species that are not easy to spot directly. Now, this is the picture which is showing the pellets of birds which have been collected, and the biodiversity scientists can recognize these pellets. Even they can sometimes they get bones from them, and this is an indirect method by which they can identify the species. Now, the modern technology provides a new technique to understand which species can or which species lives in a particular territory. For instance, photo trapping. Photo trapping is used in sampling various kind of nocturnal animals. Now, in this technique, there is a camera which is used in a remote way. So when you place the camera and you wait for the night, so that the camera can take the pictures at night, The camera is a kind of infrared sensor that catches the animals, and then when they pass through, they just get the photo of them, and by looking at those photos you can recognize what species live in that particular place. Radio telemetry is another system where if you are following the same species of the same individual for a long time, you can use this particular sampling technique. So you have an antenna which monitors the movement of the species. Other techniques like ranking and marking, usually this is used for birds. In this case, what you do is you can mark and recapture the animals to understand the dimension of the population. Now, nest counting is another system often used to monitor birds of a particular territory. For aquatic vertebrates, there are other sampling methods. One of them is the direct method to recognize and to identify species of whales. Now, this is called whale watching that you can easily do from a boat. There are other simple techniques like using plankton nets. Now, plankton nets are just nets that you use to collect plankton. They have a very thin matrix, so you can use dense matrices and vary the density or the thickness of the matrices to correct the different variety of planktons which you are interested in. Drag nets are another type of collecting aquatic species. Now, these drag nets are those that move on the bottom or of the seaside, and when you collect the species, you can take it out and identify them and make a sample out of them. You can also use another type of net which is called the ground deep net. Now, deep nets are similar to this drag nets, but the deep net are useful to measure the population of those species which live in the deeper or the interior of the sea. So in this case also, by collecting the samples through a deep net, you collect the species, identify them and recognize them. Radio detectors are very useful to detect some aquatic species. Now, if you want to understand indirectly which species, for instance, whales or dolphins live in a certain ocean or sea. Using radio detectors, you can understand which species is emitting a particular sound and their calls can be detected. There are some direct methods like using cages. Cages are very useful when you want to trap animals. In this case, you just drop, you wait, and you get to identify after the animal is caught in the cage. Rovers is a very important technology that has been developed recently. Now, these rovers are radio commanded. You can use them to go into the deep sea. So, in many cases, you cannot understand the species if you go into the deep sea using submarines or some other equipment or the divers who dive into the deep sea. But robots can do that, and robots are very useful which can have pre-programs within it, and they can use those programming to identify species. Now, there are of course systems that are more easy, like the rover, which actually onboard monitoring cannot tell. I mean, if we do a onboard monitoring technique when we go for a ocean trip, we cannot detect many of the species. counting of turtles, for instance, if you want to do that, you can walk along seaside and count the number of the turtle nest. On the nest there are turtles that are there are some techniques which can be placed, like that some kind of detecting meters can be placed on the nest to detect them. Now for direct observation becomes very difficult in certain cases. It's not easy to observe certain species directly and identify them. So some innovative methods have come up. We'll discuss it in the discuss it gradually. Now more species, there are many species we find which are very similar to each other. Even butterflies, which are so colorful, are not very easily identified. It needs a deep knowledge about the species. So using pitfall traps, nets, light traps, for instance, like some other traps like lure traps, or analyzing the soil samples, you can understand what is what is there in the soil, or sometimes by seeing the small species in the soil or the seeds found in the soil, you can identify the particular species which is found in that fauna. Now there are another very good trap which is called the entomological umbrella. It is very similar to a normal umbrella which you put under trees. For instance, you shake the tree and all the animals fall down. You can collect these animals inside the umbrella. This is a simple tent-like structure like that of a camping tent. Now that is open everywhere, but there is something that surrounds, so that animals can go inside from one side and enter inside this kind of a tent, and you can easily catch them and collect the animal and identify them. There are other systems like a bottle with funnel, Winkler trap, Niskin bottle. All the system use special traps with lights. Sometimes they have nets or small other traps to catch tiny animals. They are collected and then they are identified. There are other systems such as the Malaise trap that collect samples, and they look simply like the camping tent, and samples can be collected in various places. Now this is a picture of a Malaise trap, which is a large tent-like structure used for trapping, killing and preserving flying insects, particularly Hymenoptera and Diptera. The trap is made of a material like terylene netting and can be of various color. Now this tent-like passive intercept traps primarily, as I said, flying insects. They are set in contact with the ground to collect low-flying species. They also catch some insects that emerge from the ground below the trap or that climb up from the ground. Now, as I said, this is the picture of the trap that collects sample of mud from the ground, and along with the mud, the species are also collected which can be identified, and the sampling can be done. Now, a very simple system that is used to identify the species above the ground level use just simply by sampling squares. Now you just put the square and then you count the number of individual species which are present inside. Now in case of plants, suppose to sample vascular plants, usually plots, quadrates or transits are used. Now, as I told you, a plot is just a simple space that we identify and then we put randomly over the area that we want to analyze or study. Now, quadrates are very similar but are very useful to identify and to collect samples and species in small parcels. Now, we use quadrates for plants, for instance, but we cannot use quadrates for trees. Transects are very useful when we want to put a straight line and follow this line and identify what is there along this line. Usually, it's very important to set a priority, a kind of minimum uh, diameter of the tree, or what we call the diameter at breast height, is actually considered to be the standard for measurement of trees, and we call it dBH. Now, this is the uh, you should keep in mind that what's the limit of your sampling, because sometimes you want to collect all species that have a certain dBH or a diameter more than 5 centimeters, or it may be more than 10 centimeter. So it's better to specify when you are reporting your results which kind of dBH or diameter you are using while collecting the samples of the tree species. When you want to sample small organisms like lichen, fungi, algae, etc., you can use quadrates in the ground. Now, I told you this is very useful for small space. That is, if you want to take the sampling method by quadrate uh, or transect, you choose smaller areas. So you make an artificial quadrate, you place in a particular space, and you can easily identify the particular fungi or lichen present in that place. For instance, you can use this quadrate that's on the trunk of the trees to identify mosses on the ground, on the grass, or over the stones. That depends on which kind or which group or what's your target group. This kind of a technique is very useful. You can put random at the distance, randomly at certain distance, or you can make it a fixed, uh, place it at fixed places. For instance, like average 2 meters, or you can say that your quadrate will be of 2 meters, or you can randomly measure the identify the species at random distances. So it is up to you, or it's the sampling technique you are following will determine or decide what kind of uh, quadrate you will use. Now, another very useful equipment which should be carried while doing biodiversity studies or sampling of biodiversity, that is lens. So you need to use some lenses. It may be in the form of a microscope or portal microscope, small microscope. You should always carry for identification. Now, just to identify the species better, sometimes you need a very accurate identification to monitor and sample microorganisms. There are different systems. Of course, you cannot do this type of study with your own naked eyes. So you need, for instance, petri dishes. First, after collecting the sample in some tube, you bring it to the lab. You grow this species in a particular media present in a petri dish. So you need to grow and then allow this species to develop, and then you can identify them better. Also, you need microscopes if you want to identify some invertebrates, microinvertebrates, uh, which are present in water or some muddy soil. You cannot identify them with the naked eye, so you need this special instrument. So you need to collect your sample to and then grow in a laboratory to identify the species. Of course, this is very specialized work. You need to be an expert in doing this. And finally, I would talk about another system which has come up very recently, which is called the BioBlitz. Now, this is very specialized work, and um, it has been developed recently. It is very interesting because it involves many people and many groups in the society. It is a 24-hour job in which teams of volunteer scientists, families, students, teachers, and other community members work together to find and identify as many species of plants and animals, microbes, fungi, etc., and other organisms as possible. Now we will talk about qualitative measures of biodiversity. First of all, we need to ask what is the purpose of the research, because we can have different ideas on what we want to collect and for which purpose. One of the main reasons of qualitative research, in fact, is to increase scientific understanding of the phenomena. More interest from the theoretical point of view, which will help in proposing new hypotheses. Now, it is critical to use quality research as the basis for your experiment. But there is a great need to understand how and when to reach the formulation of hypothesis. Experimental theories that can be tested using quantitative approach explicitly. The second purpose can be to reach a deep understanding of the problem. Qualitative research has an important value for investigating complex and sensitive issues. For example, when you are interested in understanding not only the composition of the communities but also the relationship between species, like symbiosis, cooperation, etc. In these cases, it would be hard to develop a quantitative method which will not lead to more than a series of values to simplify these relations, despite the quantitative approach is predominant. So you need a qualitative method. In this type of study, where you want to describe any kind of relationship, if the aim is to try to reach a deep understanding of how species behave in a community and the role of biodiversity in the ecosystem, the qualitative analysis is very essential. The true purpose could be to provide detailed information, albeit in smaller quantities. Qualitative results certainly excel in gathering information in great detail. Of course, there are detailed quantitative studies, and many of the qualitative studies also, because they provide a collection of many numerical data. But in detailed quantitative research, such data tend to limit the analysis. The detail provided by qualitative research appear at the same time a blessing as well as can also be a diminution. What is positive is that this approach allows to describe the phenomena of interest in great detail, including aspects impossible to describe through numbers. In fact, some of the best qualitative research is often published in book form with the style that approach almost a narrative storytelling. Because most of the sector of scientific journals are careful not to propose something that deviates from a certain scientific method. The most negative aspect of quantitative research is that when this level of detail is available, it is difficult to determine what are the processes that can be generalized. In fact, many quantitative research don't mind to generalize, but simply they create detailed description of the phenomena of the study. So let's see how to conduct a qualitative research on biodiversity in practice. Now there are different methods such as field observation by the direct experience of the environment to be studied by adopting a procedure of narrative description type. A second method can be analysis of the experiences and perceptions of other or the population living in the site of the study. The third method can be the acquisition of the information derived from indigenous groups. For example, plants with medicinal use, relationship between animals, ecological functions, etc. Efforts are needed can be the recognition of the fact that the human responses to different environmental conditions are often based on the perception rather than on predominant variables, and this information can be integrated in the understanding of biodiversity trends. Addressing these issues and figuring out how to integrate the quantitative data and the qualitative information in a science-based assessment becomes easier when it is recognized that both approaches can complement each other and together reach the assessment of the result. A growing number of case studies makes use of successful combination of technical, scientific, quantitative facts, figures, tables, data, and qualitative description, perception, etc. approaches to assess biodiversity. A fifth recommended approach for the collection of qualitative information could be the compilation of a table showing on a worksheet to be used in the field the to by giving the following information. For instance, mutual relationship between plants, mutual relationship between animals or between animals and plants, antagonistic relationship between plants, between animals or between plants and animals. If some species appear to play a key role, so which, how and why you can determine, or if some species seems fundamental in considerance of the ecosystem, which, how and why questions can be answered. So describe and write the list of the species which play a role, or if some species seems to be evolutionary, co-adapted or co-evolved with each other. So they try to find which, how and why, or if some species, for instance, appear to play key roles in the study areas while others are only redundant or play a supportive function. In other case, we can take notes if some species appear to be more resilient to the effect of human disturbances in the study area, while others are more resistant, or others are can completely disappear. There are many variations to this information which can be adapted to the type of studies that we want to carry out and particularly to the objective of the research. Now, this was a general review of the qualitative research that we can use to analyze our study area and to merge with our quantitative data. In this case, we have a more complete information that can be available for different studies and different approaches. So thank you and see you in the next lecture.